Hello everyone. After first experiment, then your experiment number 2, that is to understand the method of section cutting technique, staining, mounting and observation of section under the microscope. Which thing we have to study in your experiment number 2, that is your cutting techniques, how you can cut your section. Then staining, that is which type of staining reagent you have to use. Mounting and observation, that is how you can prepare your slide for the observation, that is observe under the microscope. These three things we have to study in this experiment number 2. Then preparation of sample for the sectioning, that is you have to prepare your sample for the sectioning. Then new concept, first one is your transverse section. Transverse section is obtained by cutting along the radial plane or the cylindrical portion of the stem, root, stolon and perpendicular to long axis. This is nothing but the transverse section. ट्रान्सफर सेक्शन आपल्याला कसा घ्यायचा आहे त्यांनी त्याच्यात सांगितलेलं आहे. नेक्स्ट इज युअर स्टेनिंग. टू डिस्टिंग्विश द अरेंजमेंट ऑफ व्हेरियस टिश्यू in the samples of crude drug, chemicals, that is you have to distinguish different types of the tissue by using the staining reagent, that is your chemicals, dye or colorants are used to impart color to various tissue in the section of the drug sample. Next is your observation. You can observe this slide under the microscope, that is different into the laboratory where the sufficient light is available is important. Low power observation helps to draw schematic diagram for distinguishing transverse section. High power observation is to be used, that is you have to observe your slide where there is a proper light. In this way you can do your job, that is your observation. These three things are important and all these three things are very important for your experiment. In each experiment there is a sample preparation, then transfer section, then staining and then observe under the microscope. Then for next is your which things you have to required, that is first one is your microscope, that is your microscope. Then next is your watch glass, this is your watch glass. Okay, next is your camel hair brush, this is your camel hair brush. Next is your glass slide, this is your glass slide. Then cover slip, then beaker, then dropper, then filter paper, then forceps, then test tripod stand, wire gauze, dissecting needle, then your sharp razor, etc. These are the important things which are required for the section cutting. You can see in this way, that is these are your important things, that is your microscope, then your brush, then forceps, slide, cover slip, sharp eraser, then watch glass, then your dropper, forceps. These are the important things for your section cutting. Okay, then after that procedure, section of a stem, root and stolon, that is different sections can be obtained from the root, stem, stolon, etc. that is depending upon the plane of cutting. Each section revealing details for the different angle, that is section you have to take in this way, that is your longitudinal section. Transverse section is obtained by cutting along the radial plane of the cylindrical portion of the stem, root, stolon and perpendicular to the long axis. This is your long axis and this is your radial plane, and then you can take your section. Okay, this is about your TS. Section when prepared and observed under the microscope reveals the radial arrangement of the tissue and shows concentric layers of the vascular bundle. Then after getting your section, these things you can observe under the microscope. Then section of the leaf. लीफ जे आहे लीफचा सेक्शन तुम्ही जो आहे दॅट इज आस्पेक्ट स्टडी थ्रू द मिड ड्रीप. दिस इज युअर लीफ. ओके, दिस इज अबाउट युअर लीफ. देन दिस इज देअर मिड ड्रीप. हे जे आहे मधलं सेंटर, मिड म्हणजे मधलं मिड सेंटर. देन यू हॅव टू कट the section in this way. You have to cut it properly. This is about your section. Then next is your section of the bark. This is revalence, horizontal cells and the radial. Then next is your section of the fruits and the seeds. This is done by the generally TS of the various parts is observed under the microscope. In case of fruit and seeds, the separate section techniques is required for individual drug. This is about your different types of the parts of the drug, that is your root, then your leaf, then your bark, then your fruits and seeds. We have to take different type of the section cutting technique. Mostly all we use that is your transfer section. There are two types of the section cutting, that is your TS and longitudinal section cutting technique. Then section cutting techniques, these are the main important things. Materials is to be required for the section cutting, that is your napkin, dropper, filter paper, stain, drug sample, forceps, test tube holder and stand, needle, camel hair brush, watch glass, sharp eraser blade, micro slide and cover slip. Again you can observe this thing, that is these are the requirement for the section cutting. Okay, then next is your section cutting can be done by taking transverse section as well as longitudinal section. Mostly the transverse section is taken for study of the crude drug. Presently I have also told you that is there are two types of the method, that is your TS, ALS. TS, that is your transverse section, ALS means your longitudinal section. But mostly for the crude drug we have to use that is your transverse section method. This that is section cutting is skill which facilitate easy learning of the tissue component. By this method TS you can easily understand the tissue component. There are various techniques of section cutting depending upon the part of that drug, that is which part you have to use. That is if you have to use this bark, then if you have to use leaf, there are the different types of the techniques you have to use. Then next is your, that is this leaf you can easily cut with the help of your sharp eraser. Okay, in this way you can easily cut, yes or no. Then you have to take the section of the leaf. This easily you can cut. Why you can easily cut? Because it is very soft. You people, that is next is your, this is your bark. This you cannot cut easily because it is very hard. Therefore for the technique, that is in our aim we have to see that is a section cutting techniques. For that purpose section cutting includes these following steps. That is first one is the preparation of sample for the section cutting. That is this is very hard, yes or no, nobody can cut it easily. Therefore first one you have to take these samples, that is boiling is the first step. That is you have to take the sample into the beaker. Okay, then that boil the sample, that is you have to boil the this way, that is your first, that is boiling the sample. Okay, this is after boiling, then you can take the sample into the watch glass. Okay, then section cutting, then you can cut your section. In this way you have to cut the section. Okay, then transfer the section into the watch glass containing water. इसके बाद first is your boiling, then cutting and then transfer into the watch glass containing water with the help of brush. This is your फर्स्ट प्रिपरेशन ऑफ सैंपल। अभी देखो ये जो सैंपल था ये बहुत हार्ड था, this sample is very hard, therefore we cannot cut it easily. Therefore first procedure is the boiling. After that there is a cutting the section. Okay, after cutting then transfer the section into the watch glass. Okay, next is your procedure जी आहे तुमची staining. आता what is mean by staining? First all of you know what is mean by the staining. That is, stain is a chemical dye or colorant which combines physically or chemically with a cell containing to impart color to it. For example, Sudan red tea dissolves in the fixed oil present in the oil seeds to impart red color. That is, first all of you know that is what is mean by the stain. Stain means जो है that is your dye which combines physically or chemically with a cell. That is, all of you know white dresses which is make color by the laundry. This is the same, this is your dye or the colorant. In this staining procedure how you can done, that is, take a clean watch glass. आप लोग क्या लेंगे, this is your clean watch glass. Okay, and add staining solution to it. Then after that to this add, this is your staining solution, add to into the watch glass. After that with the help of brush transfer the section taken from the watch glass containing water to stain solution and keep it for 2 to 3 minutes. That is, we have to transfer this section, okay, with the help of brush into the staining region, that is second watch glass that's containing the stain, and keep it here for 2 to 3 minutes. Then transfer to the watch glass containing plain water so that excess staining is to be washed away. This section is ready for the mounting. If we have to put the section here for or more than 2 to 3 minutes, then this transfer into the another watch glass containing water for removing excess of staining, and this section is ready for the observation. Then you can transfer this section onto the slide. In this way you can transfer this section onto the slide. Is then transfer the section to mounted on the watch glass with the help of brush. Then after that add 1 to 2 drops of water on the section with the help of dropper. This is, in this way you can add 1 to 2 drops of the water on that section with the help of dropper. Then place a clean proper cover slip over the section with the help of forceps and needle. Next procedure क्या है आपकी, that is with the help of forceps. This is your forceps. Okay, this is your forceps. Then this is your cover slip. With the help of this forceps you can transfer it on this section with the help of forceps and this needle. All of you can see this is your needle, this is your forceps, this is your cover slip, and in this way you can put it here on the section. Okay, then if there is a blotting, if there is a excess blotting here, then swap it with the help of filter paper. In this way this is your slide which is ready for the observation, and then keep this slide here on the stage to observe the section. This is the simple method to observe. In this, next is the important thing, that is this procedure described above the routine laboratory technique. So there may be evaporation of water and the slide paper will not be last long. Glycerin is used to avoid evaporation of water and drying the section. If here we can add which thing, water. All of you know, with the help of dropper we add water, but water it is the evaporating. Then due to the evaporating, you people add which thing, that is your glycerin. glycerin क्या करता है? glycerin जो है वो सेक्शन को ड्राई होने नहीं देता. This is about the main reason for the avoid the evaporation of the or dry, to avoid the dryness of the slide. This is your routinely procedure you can do into experiment number second. Complete your experiment number second. आता तू ह्या एक्सपेरिमेंट बोल.